Welcome to the second presentation on molecular genetics. I'm Wendy Nofke and I'll be doing this brief presentation for you today. Today we're going to look at protein synthesis. Protein synthesis is the process of making proteins, and it's going to include two subparts, transcription and translation. Transcription is going to be the portion that occurs in the nucleus. It's going to use the DNA to make an mRNA. Here you can see in this example here you've got DNA, and it's being used as a template to make this mRNA here in the nucleus. During translation, the ribosomes are going to use that RNA in the form of mRNA to make a protein, and that will occur in the cytoplasm. Transcription is the portion where you use the DNA as a template to make RNA. The mRNA is complementary, and it's made from just a single strand of DNA. So at the beginning we have the initiation of the process where an RNA polymerase enzyme is going to bind to a promoter region of the DNA. Here it will unwind the DNA and expose the base pairs on the inside for the enzyme to be able to get in and read the sequence. During the elongation phase, nucleotides are added to the mRNA strand. And then in the termination phase, the mRNA strand is completed and detaches. So as you go along here, you can see nucleotides are continually being added on. Termination does not occur until it's actually reached the end of the strand. After termination, that mRNA is going to be ready to be modified and sent into the cytoplasm. So in a eukaryote, the mRNA does need to be modified before it goes into the cytoplasm. Some of these techniques include splicing, editing, and polyadenylation. With splicing, you're going to remove the introns or intervening sequences and leave the exons or the express sequences. Eukaryotic DNA has these regions where some of it is going to be expressed and some of it is going to just be placeholders in between these express sequences. And the express sequences are your exons, the intervening sequences are the introns. Editing can occur, and that will change some of the nucleotides to make different forms of the protein. With polyadenylation, a tail is added on of adenines. This will help to signal the end of the gene and also aid in transporting the mRNA. With the genetic code, it's created by a sequence of bases, the A, T, or U, and then G and C. We have 20 different amino acids and 64 possible codons. A codon is a group of 3 base pairs that will code for a specific amino acid. We need to use 3 base pairs in order to have enough possible codons. If we only used a single base pair, that would only allow for 4 different amino acids. If we use 2, that would be 4 times 4 is 16 possible combinations, still not enough for all of the amino acids. So we use 3 of them, so it comes out to be 4 times 4 times 4, which is 64 possible codons. This genetic code is going to be redundant because there is only 20 different amino acids and 64 possible codons, but it is unambiguous. This is an example of a codon table. Rather than memorizing what amino acid is coded for by each codon, these tables are used to be able to quickly read that. The first letter in the codon is found here along the left side. For example, they all are going to start with AUG, so we can find the A here. The second letter in the codon is found across the top. So if we take and come down to the box that is at the intersection of the first 2 letters here, we can see we have A and U. Over here on the right we find the 3rd letter, and this corresponds here with AUG, which codes for methionine. So the same process is used to determine what amino acid is going to be placed at any given location based on what the codon is. When we read the code, it starts at the first AUG. This codes for the amino acid methionine, but it's also going to establish the reading frame of every codon down the line. The mRNA reads codon by codon until it reaches the stop codon. There are 3 codons that are considered stop codons: UAG, UGA, or UAA. Both of those do not code for any amino acids and therefore signal the end of reading. This code for reading the codons is nearly universal. During translation, you have the process of the RNA being read to make the protein. The ribosomes are going to read the sequence of the codons on the mRNA. As they do, it will use the tRNA or transfer RNA to bring the amino acid to the ribosome in the correct sequence. Each tRNA has a portion that is complementary to the codon called the anticodon. This way it's able to make sure with complementary pairing that it's brought the correct amino acid to go next in series. As the amino acids are brought down to the ribosome, it will bond to form the polypeptide chain. Then the ribosome will move to the next codon and the tRNA will be released and recycled for future use. After the polypeptide is formed, it undergoes additional processing, such as folding or combining with other molecules, so that that protein can go off and do its proper job in the cell. This is an illustration here of the peptide synthesis. Down here we've got the messenger RNA, or your mRNA. Here you have the ribosome that is reading the code. You've got the tRNA that is bringing down the amino acid. You can see here that there is A and U, G and C pairing, that the tRNA is going to have its anticodon bind in a complementary fashion to the codon. As it does that, you will have the amino acids attach, and they're going to form a line that's going to hang behind as you move down the line. This allows for very accurate reading of the DNA.